Okay, so let's take a look at the big picture here. So transcription is making a copy of your information, the particular information that you want. So that's like transcription happens in the nucleus. So that's like the cookbook in my office. The copy, the mRNA, is made in the office and brought to the kitchen. And finally, making the recipe is the translation, taking those instructions and turning them into something useful. So if we want to, again, compare that to DNA, your DNA is stored in the nucleus. Your mRNA is made in the nucleus and brought out into the cytoplasm. And then your protein is assembled in the cytoplasm. Okay, and making a protein is called gene expression. Okay, you're translating the instructions into proteins. Okay. So each cell type expresses some genes, but not others. So that's what makes them unique from one another. Okay, so that's why a heart cell makes pounding heart cells, where liver cells produce these detoxifying cells. Okay, and this is what an mRNA molecule looks like. So if we're looking at the DNA, we're going to look at the piece of DNA that we actually want to produce here. Okay, so when you transcribe it, you're going to make a complementary strand to the strand that you're seeing. So our original DNA template strand, it had ACC. So it goes, and remember now, instead of T, A is going to pair with U. UGG. Okay, so AAA is transcribed into UUU. CCG to GGU, C, and AGT to UCA. Okay, so you're not making a straight copy, you are actually making a complementary copy. Okay, so let's try this. Okay, so we're going to be making a complementary copy. So T is complemented by A again, and then A is complemented by U and mRNA. G is complemented by C, G, G, U, C, G, 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 C, A. Okay, so that's my mRNA strand. Now here, we're going to do the same thing. So A is going to be complemented by U. We're going to get C, G, then 3 G's in a row. C, G, A, C, C, A, U. Okay, so that's just transcription. That's taking your DNA in the nucleus and copying it so that it can be taken out into the cytoplasm to be made into something else. Turn the page. There we go. So it moves to the cytoplasm so it can make a protein. You read 3 nucleotides at a time. So that's what is read when the DNA is translated into a protein. So 3 nucleotides in a row, 3 nucleotides are a codon. So that's like a word, okay? And it translates into an amino acid, which is the language of proteins, okay? And if you think about amino acids are the monomers of proteins. So if we're looking at a pearl necklace, the monomers are the pearls and the necklace is the polymer. So proteins are the polymer made up of the amino acids, which are the monomers. Okay, so we're going to read these 3 letters at a time. So mRNA is translated into amino acids using the codons, and they're joined together to make proteins. And I know what you're thinking, but where did you get these amino acids from? Well, there's these charts called the codon chart. Okay, it's the genetic code. You read it in groups of 3. Okay, and it's the instructions for a protein. So there are 20 different amino acids, and you use a table that you'll always have available to you to translate the mRNA sequence into amino acids. Okay, because there are 3 letters and they can combine in a bunch of different ways. Some codon words mean the same thing. Some words tell you to start making a protein, some will tell you to stop. Okay, so this is a genetic, this is called the codon chart. So the way you use this, let's say that you have the amino acid sequence, the mRNA sequence CAU. What you do is you start here on the left-hand side where it says first nucleotide, and you look in the C column. So you're going to look, first nucleotide is C, so I'm going to be looking in this column here. Okay. Now I'm going to go right here to my second nucleotide. My second nucleotide is A, so I'm going to go down to A. It's going to put me in this box. And my third nucleotide is U, right? And if I erase all this stuff that I have here, it tells me that my amino acid is going to be HIS, which is his, which is short for histidine. Okay, so let's try this. All right, I have the universal chart right here. So TAC. So what I'm going to do, which is helpful for me, I'm going to draw a line every 3. Did I mess up? I did not. Okay. So remember, our mRNA is our complement. So T is going to be A, U, G. I'm going to look at my chart. My first base is A. Okay, so I'm going to be looking in this area here. My second one is U. So I'm going to be looking in this block right here. And then AUG means methionine or start. So my amino acid is MET, M-E-T. Okay. GGC, I'm going to do CCG. I'm going to erase this. So first base is C. Second base is C. So I'm going to be right in here. And look, all 4 of these actually code for proline, P-R-O. C-A-T. I'm going to erase all these. So first base, C. I'm going to be right here. A is over here. So I'm going to be in this. Oh, you know what I did, guys? I did not translate it first. That's my bad. So I'm going to do G, U, A. So I need to go to G, U, A. So first is G. So, and second is U. So I'm going to be in this box here. GUA is Val. Okay. And then next is CAG, so it's going to be GUC. GUC gets me Val again. I'm just going to fill this in for you. GCA gets me CGU. It's going to get me arginine, ARG. UAU. Now look at this, guys. Oh wait, not yet. UAU is serine. Okay. And you know what? I'm missing a letter here. The letter G here. So CTG. That gets me GAC. And that gets me asparagine, ASP. Okay. I want you to try this one on your own, so pause the video. Okay, and I'm going to fill it in real quick for you. Okay, so pause the video, try doing it on your own, and then I will put the answer up real quick, okay? Remember, try to space them out. Okay, here's the answer. And then this next one. Remember, you could pause this video at any time if you want to see something again. So we've got, you can pause it now and try to transcribe and translate this on your own. Oh, I'm doing the wrong thing. I apologize. I'm filling this in incorrectly. So when you hit a stop sequence, your entire protein stops. So this does not keep going. Okay? No more amino acids. Once you hit a stop, so if you look here at UUA, I'm sorry, UU, it shouldn't be UU. Oh, you know what? It should be UUG. So pretend this was a C. UUG. When you hit a stop, that's it. The whole thing ends. All right. So you start with DNA, you transcribe it into RNA, and then you translate it into proteins. That is the central dogma of biology. If you have any questions, feel